Plants grow with the tip of the roots and shoots, otherwise known as growing points. But do humans have such growing points? If not, how do we grow? For a multicellular organism to grow normally, the cells must divide. The cell division process that takes place is called mitosis. You may now wonder, what is mitosis? Mitosis is a form of nuclear division that produces daughter nuclei containing the same number of chromosomes as the parent nucleus. The daughter nuclei are genetically identical. In simpler terms, mitosis is where the DNA of the cell nucleus is split into two equal sets of chromosomes. The cell division takes place in many stages, and the order of events is known as the cell cycle. The interphase is where the cell spends most of its time, but not to worry, although it is a resting stage, the cell still absorbs nutrients. At the interphase, the chromosome appear as thin long threads called chromatin. Before the cell divides, each thread replicates, producing two identical DNA molecules joined at the centromere. In the prophase, chromatin threads condense, coil, and shorten to become chromosomes. Each chromosome consists of two sister chromatids attached at the centromere. Then come the metaphase, where chromosomes line up along the equatorial plane of the spindle. The centromere of each chromosome is attached on both sides to a spindle fiber. Next is the anaphase. This is where each centromere divides. The spindle fibers shorten and pull the chromatids apart to opposite poles of the cell. Once chromatids are separated, they are called daughter chromosomes. This is followed by the telophase. Spindle fibers break down. A nuclear envelope forms around the chromosomes at each pole of the cell. Then, a nucleolus reforms in each nucleus, and the chromosomes uncoil and lengthen to become thin chromatid threads. Lastly, cytokinesis occurs. This is the division of the cytoplasm. In animal cells, cleavage or furrows appear in the cytoplasm between the two nuclei. The furrows deepen, and two identical daughter cells are finally produced. The first checkpoint is the G1 checkpoint. This checkpoint is the main decision point where the cell must decide whether or not to divide. Once the cell passes the G1 checkpoint and enters the S phase, it heads irreversibly down the division pathway, producing two daughter cells. However, the cells must first fulfill specific conditions to pass the G1 checkpoint. The checkpoint assesses the cell size, whether the cell is large enough to divide, the cell's nutrients, whether all needed nutrients are present, and let's say the cell does not get the go-ahead at the G1 checkpoint. It will then enter a resting stage called the G0 phase. Moving on from the G1 checkpoint, the cell has an additional checkpoint before the onset of M phase to make sure that the cell division goes smoothly with no damaged DNA. This is called the G2 checkpoint. At this stage, the cell is mostly concerned with the condition of its DNA. The DNA of the cell will be checked with concerns such as DNA integrity, whether any of the DNA is damaged, and DNA replication, if the DNA was completely copied during the S phase. If damage are detected, the cell will pause at the G2 checkpoint to allow for repairs. If the problem is successfully fixed, the cell will re-enter the cell cycle, proceeding normally with cell division. If the damage is irreparable, the cell will undergo apoptosis, also known as programmed cell death. This self-destruction mechanism ensures that damaged DNA is not passed to daughter cells. Now the cell has successfully entered the M phase. Its current goal is to split its duplicated chromosomes evenly and accurately between its two daughter cells. This will take place at the spindle checkpoint. The cell confirms the alignment of the cell if all of the chromosomes are lined up at the metaphase plate, and the cell's tension, whether each chromosome is under tension from the spindle.